Good morning. I'm sorry you had to get up early to come and hear me on a Saturday. I'm going to give this talk, What is Life? It is based on my book, as you just heard, but it is built around 5 great ideas in biology, so it fits with this session. But before I start, I just wanted to say something about these ideas, because they are ideas that you will all know already. And that doesn't mean that they're not extremely important and interesting, and indeed, maybe you've learned quite a lot about them already. But they are extremely important because they underpin biology. And if you listen carefully, you'll probably find out some things you didn't know about, like where they came from and how they all fit together. So just because you think you know all about it, it's just possible you might not. Okay, so that's just I wanted to say. So the title of this talk is What is Life? And it's one of the most fundamental questions in biology. In fact, you could say it is the most fundamental question in biology. Very easy to ask, not so easy to answer. Look at life around you, the extraordinary diversity of life on our planet. Bacteria, fungi, plants, animals, and of course, ourselves, human beings. And what this question is asking is, what is it? What is it that links all these different organisms, from a bacteria, a virus, to you? What is it that is common about all life on this planet? It's a long time ago, but I think it was this butterfly, not this particular butterfly, but this species of butterfly that you see behind me, that first started me thinking about these sorts of problems. I was a bit younger than you, maybe 11 or 12. It was early spring, and I was sitting in my garden in northwest London, near Wembley Stadium, which some of you may have, of course, heard of. And I was sitting in the garden when this quivering yellow butterfly flew over the fence. It turned, hovered, briefly settled, shadow disturbed it, took flight, disappeared over the opposite fence. Nothing very exciting. But that intricate, perfectly formed brimstone yellow, that's what it was, brimstone yellow butterfly made me think. It was both utterly different to me or to you, but somehow it was familiar as well. Like me, it was obviously alive. It could move, it could sense, it could respond to the environment around it. It seemed really full of purpose, purposeful behavior. And I found myself wondering, what does that really mean? What does it mean to be alive? And that is, of course, the same question as what is life? Now, I'm not the first to ask this question. It's been asked by quite a few others before. And you'll see this is a book, a very important book, written in 1944, also called What is Life, by the famous physicist Erwin Schrödinger. He was a physicist, but he wrote one book on biology, and that was this. It was influential for me. I read it over 50 years ago. And I recommend you have a look at this book as well. I don't agree with all of it, it has to be said. I think he got some things wrong. But it is built on, my response to it was to write a book that focused on these 5 great ideas. And that's what I'm going to do today. I'm going to describe where they came from and be surprised that most of them are quite old. They've been around for quite some time. And then at the end, I'll pull them together to develop principles that are fundamental to understanding life. So that's what we're going to do. My first idea is the concept of the cell. The cell is the basic structural and functional unit of life. I like to call it life's atom. It's the basic unit of all life. They were discovered in 1665, first observed by Robert Hooke in the city in which I live, actually, which is Oxford. And this is the sort of microscope he used. And the image in black and white that you see there was the image that he drew. What he did is he took a razor, he cut a thin slice out of a piece of cork, looked at it under the microscope, and drew this picture. And you'll see I put a modern picture, which is exactly the same, really. We haven't seen so much advance in 3.5 centuries. Exactly the same. And he saw neat arrays of boxes. He called these cells after the Latin word cella, for small cubicle. And he published it in a very famous book called Micrographia, which is an amazing book of the time, where he made lots of images taken from under a microscope. Now, a few years later, a Dutch draper called Anthony van Leeuwenhoek, who was working in Delft in the Netherlands, scraped between his teeth and discovered single-celled life. Now, he sent this picture, it's one of 1,000 letters that he wrote to the Royal Society in London, which had only been in existence about 20 years. It's a rather charming image. If you look at figure B, he was obviously looking at under the microscope, and you'll see this bacteria was swimming around. He did a loop. Can you see there, going from C over to D? He was rather disturbed by this discovery of finding living microorganisms living between his teeth, because he was very proud of his dental hygiene, and discovering life in his teeth upset him a bit, it has to be said. Now, he was a close neighbor in Delft, that's a city in the Netherlands, to a painter some of you may have heard of called Vermeer. Vermeer is a famous painter, and he, very unusually, painted 2 paintings of the same person, a scientist, a geographer. He called one an astronomer, another. This is one of them. It's the one in the Louvre in Paris. And I like to think this is Leeuwenhoek. There's no evidence for that, really, except for one thing. He lived in the road, the street next to the one where Vermeer and Leeuwenhoek lived in adjacent streets. And when Vermeer died, he died in his 40s, Leeuwenhoek looked after his will. So maybe they knew each other, maybe this is Leeuwenhoek, maybe it's not, but it's quite nice to think it might be. Now, over the next 2 centuries, it became clear that all living things were either consisted of a single cell or a collection of cells. We are a collection of cells. And it was well expressed by this German professor, zoologist Theodor Schwamm, who in 1839, in German, this is a translation, said, "We have seen that all organisms are composed of essentially like parts, namely of cells." And this essentially says that the cells are the basic structural unit of life. But they are more than that. They are also the basic functional unit of life. And that was well expressed by another German scientist, Rudolf Virchow, who in 1858, so about 20 years later, but notice this is over 150 years ago, stated every animal, you thought about animals, but it applies to plants and fungi as well, every animal appears as a sum of vital units, each of which bears in itself the complete characteristics of life. This means, of course, that it's the vital unit, complete characteristics, it's the basic functional unit of life. He also said something else. Omnicellular acellular, this is a Latin for all cells come from cells. And this means cells arise through the division of pre-existing cells. And that was contentious at the time. Some people thought cells arose spontaneously in liquids. And this said cells always had origins from another cell. And just to point out how important that is, we stretch back over time through cell divisions, right back to the beginning of life. Just think about that. we stretch right back over 3.5 billion years, connected physically by this chain of cell divisions. We don't often think of it like that, but it is true. So I hope I've convinced you that cells are interesting. If I failed to do so, have a look at this. This is a cell. It's an egg being fertilized by sperm. And I'll remind every one of you, you were once looked like this. Every one of you was once a single cell. And if you're not interested, there's doors on either side. You can leave now, because it's a really interesting point to think. So cells are considered the simplest entities which unambiguously exhibit the characteristics of life. I say unambiguously because I'm going to talk a little bit about viruses, which are a little bit in between. But cells, everybody would agree, are living. And this means understanding cells and how they work is key to understanding life. That's what I do research on, and it's why I research it, and it also will figure prominently in more things that I will say later. This is a modern picture of a cell, artificially coloured, and I'll just say a few words about what cells are. They are bounded physical entities separated from their environment, but in contact with their environment because they are surrounded by a semi-permeable membrane made of lipid. This means they're separated, but they are in communication with their surroundings. And this separation allows order to be built up inside cells at the expense of increasing disorder outside cells. And in this way, life does not contravene the second law of thermodynamics in physics, which is often an issue that troubles physicists thinking about life. They get agitated about the second law, but there is no contravention of the law. Even Schrödinger got a bit exercised by this. He said, an organism's astonishing gift of controlling a stream of order on itself and thus escaping the decay into atomic chaos. This is a picturesque way of talking about the second law of thermodynamics. He was astonished that that could happen, but it led him to propose that life was based on a code script. He wasn't the first to do that, but he emphasized it. What he said here, you see, is these chromosomes, he was talking about chromosomes, I'm going to tell you something about those in a moment, contain in some kind of code script the entire pattern of an individual's future development and function. So he proposed life had a code script. And that introduced me to the second idea I want to share with you, and that is that the gene, and all living things have genes, which can explain the mysterious phenomenon of inheritance. Now, this is the idea that the gene is the basis of heredity, which had its origins with the work of a monk in Austria called Gregor Mendel. This is a picture of him. He was abbot of Brno Monastery, and he published two important papers in 1866. I've been to Brno, I've been to the monastery, and I have looked and indeed touched his original handwritten manuscript of these two papers. I'm a geneticist as well as a cell biologist, so that was very important for me. So Gregor tackled the problem of inheritance by carrying out crosses of pea plants which have different characteristics and analyzing the outcome of those crosses in the thousands of progenies that they produce. And he had essentially in this monastery a research institute. We don't think of it like that today, but it was essentially a research institute. I took this picture in the middle of the Cold War. It was in 1981 when I visited there. And this was his research garden. It was then under communist rule. And it was a really big garden, And not only that, he had a greenhouse of the same size, which had been dismantled, but it's being rebuilt now, actually, of the same size to do his research. So it was really rather like a research institute. And what Mendel did was to cross peas with varied characteristics. Here's the sorts of different characteristics: tall plants, different colors, short plants, and all sorts of different characteristics. And you will have been taught this, I'm sure, all at school. But he famously observed simple ratios in those characteristics as they underwent segregation in the progeny. Ratios like 3 to 1, and 9 to 3 to 3 to 1, and 1 to 2 to 1. You're all familiar with this. I'm not going to try and describe it. This led to what we call Mendelism. Mendel didn't quite get it right, actually. It took another 30, 40 years to get it right. And what Mendelism encapsulates is that heredity is based on pairs of indivisible particles or factors, one of which is inherited through each germ cell, the pollen and ovules in plants, sperm and eggs in animals. It was because it's particulate that you get these very clear ratios, like 3 to 1. He, by the way, was trained as a physicist. He failed all his physics exams, by the way, in Vienna University. But he took his... So if you fail exams, don't worry too much about it, because it is okay. I failed my exams as well. And he applied, however, his physical and mathematical thinking to biology. These factors that he postulated were subsequently found to be linked to the chromosomes, which Schrödinger mentioned, which were only discovered after his death, 30 years after his death. And I'm showing you a picture here from the 1890s of an onion root tip cells undergoing division. Can you see those little strings there in these cells? And these are the chromosomes separating. I first saw chromosomes under onion tips as well when I was 12, 13 years of age. And so we were still using the same material. Now, his idea, nobody took any attention of it. It was ignored completely for 30 to nearly 40 years. And I think the reason was that his interpretation of his data was very abstract. It was based just on numbers, and it didn't sort of excite people. But this abstract interpretation was turned into a concrete one when people observed these chromosomes, because then they saw that the chromosomes behaved just like Mendel's factors. And of course, these genes are found on these chromosomes. In the 1940s, Oswald Avery in Rockefeller University in New York showed that genes were made of deoxyribonucleic acid. He did that in 1944. I worked in Rockefeller University. I was president of it for 7 years. And that led eventually, a little later, I'll explain in a moment, to the structure of DNA. So what we see here is the nucleus, which is in the cell. That contains the chromosomes. If you unravel the chromosomes, then you can see that they are made up of DNA, and you can see the famous pairing structure of DNA, which we're going to talk about in a moment. Now, this structure was established about 10 years after Avery in New York. It was established in London and in Cambridge. In London by Rosalind Franklin, Ryan Gosling, and Maurice Wilkins, who did the experimental work. In Cambridge by Francis Crick and Jim Watson. I run the Francis Crick Institute in London, named after him, who did the theory explaining the X-ray crystallography and what it meant. And Crick and Watson famously argued that the double helical structure of DNA, which you'll be very familiar with, could explain heredity through this linear sequence of bases that make up DNA and the pairing between bases. Think of DNA as a ladder. It's twisted, but it sort of makes it complicated. So in your head, untwist it and just think of it as a ladder. Then split the ladder apart, and you've got rungs. Rungs on the left side, rungs on the right side. These are made up of bases, of adenine, which pairs with thymine, cytosine that pairs with guanine, and this means when you tear it apart, then it forms a template to make an exact copy so that you can completely replicate precisely the same molecule. And you see that is the case as you can see here. And this is, of course, Schrodinger's code script. That's exactly what Schrodinger's is. Crick, who was a very, I knew Crick quite well, a very imaginative man, very innovative, went on to propose what he called the central dogma, explaining how DNA encodes the structure of proteins. And that's shown here. The central dogma describes how the sequence of bases making up a gene is copied from deoxyribonucleic acid into ribonucleic acid, a somewhat simpler structure, and that acts as a messenger between the nucleus and the cytoplasm, so it's called messenger RNA, and it goes from the nucleus, where the chromosomes are, into the cytoplasm, where that RNA encodes proteins, and that's where proteins are made, and of course proteins do all the work in cells. They act as enzymes and other things. These discoveries, which I've just described to you, laid the foundations of what we now call molecular biology, done in the 1950s, 1960s, a bit of the 1970s, and they are critical to understanding life. So I'm just going to repeat it so it's clear for you. Central to it is the genetic code. It's written in a 4-letter code of 4 bases, A, T, C, G. The information is stored in linear form. That's really important, linear form. This way of storing information is the same as the sentences I'm speaking to you, the sentences you're writing in, or some of you are writing in your notes, or you're reading in a book, or for that matter, the bytes in a computer, the digital storage, is all stored in linear form. It's the most efficient and effective way of storing information. But let's be clear about it. This way of storing information was invented by life over 3 billion years before the computer age. So we are 3 billion years late in the invention of this very critical thing. Now, the nature and function of the gene introduces the 2 next ideas I want to explain to you. Life as chemistry and life as information. Firstly, life as chemistry. Now, how does chemical activity and physical forces, of course, bring about life? The origins of this idea are also old. They can be traced back to Lavoisier. Lavoisier is the father of chemistry, worked in the late 18th century. His research was terminated because he was guillotined, actually, in the French Revolution. But he started looking at biochemistry of fermentation. But it was Louis Pasteur, who I show here. This is a picture in the Pasteur Institute in Paris, who carried out experiments that were critical. What he was doing was trying to help industrialists who took sugar beet, this was in northern France, and fermented it to make alcohol. But what the industrialists found was it quite often failed and made acid instead. And they turned to Pasteur to see if he could help them. And Pasteur looked at the fermentations that worked and the fermentations that didn't work under a microscope. And he found that when it worked, there were yeast cells present. And when it didn't work, there were no yeast cells present. And he speculated there were bacteria there, and that the bacteria were making acid and the yeast was making alcohol. So he told the industrialists, if you put yeast in this, it will always work. Whether they paid him for this, I don't know, but it certainly meant that the fermentations would then work after that. But he was a scientist. He wasn't just interested in solving an applied problem. He wanted to see what did it mean about the nature of life. And he concluded the following in French, but this is translation. Pasteur said was fermentation was a physiological act yielding products for the cell, and that chemical reactions are an expression of the life of a cell. In other words, life is chemistry. But it's a very elaborate, very special sort of chemistry. It's based on carbon polymers. DNA and RNA, I've already mentioned, are carbon polymers. That is, they are carbon atoms connected in a chain in the linear form. But now I want to talk briefly to you about the carbon polymers that are proteins. And these are the substances from yeast that Pasteur postulated made the alcohol, because these were proteins that acted as enzymes that catalyzed the chemical reactions that made alcohol. Now, proteins are polymers of amino acids, and they have a backbone with side chains. If you look at the bottom of this slide, you'll see there's 2 carbons and a nitrogen, 2 carbons and a nitrogen, so it's a carbon polymer with nitrogen too. And that's the same throughout a protein. But what is different is the side chain, because there's 20 different amino acids. And I've identified on here 3 or 4 different ones. They can be small, bulky, or they can be acidic, or basic. They can be hydrophobic. They don't like water. And what this means is that different amino acids have different chemical properties. And if you now link them together, you can produce a very diverse chemical situation, which allows the proteins to do different things chemically. And that is the basis of all the chemical reactions that go on inside our cells. So you have DNA, which is boring and constant in its chemistry. It doesn't do very much chemically, which is then turned into proteins, which are chemically active. It's a very clever solution. Here you see the structure of the amino acids, and here you see that the different chemistries mean that the chains of the protein can fold up in different ways to produce extremely different structures. And so the ability to produce different structures with varied chemistry is the reason why proteins as enzymes have such enormous diversity in the different chemical reactions that they can carry out. And this is an example of a folded up protein. Looks as if it's made up of billiard balls of different colors to reflect the different chemistries that proteins as enzymes can carry out. So these are acting as chemical machines. And these machines produce the huge range of chemicals required for life to work. They make new molecules, they break molecules down, they recycle them. They can make DNA, RNA, other proteins, lipids, carbohydrates. They can do physical work. They can make motors that carry cargoes around inside cells on protein-based trackways. They can act in hybrid ways, combining chemistry and physics, capturing energy from sunlight and turning it into energy-rich chemicals. This is the basis of the chemistry of life. But it's something extraordinary about it. A cell is tiny. It's only a few micrometers across. And within it, simultaneously, thousands of chemical reactions are going on at the same time. Just think about that. You know, you have a test tube, you've got one reaction. You've got a cell just 10 micrometers across, and there's thousands of reactions. This is remarkable. So how does this actually occur? This just gives you a taste. Each of these dots is a different chemical reaction. There's about 150 on here. Krebs cycle is that round circle for those of you who've been taught this. But there are thousands of these dots, each a different chemical reaction in cells. How does it occur? It occurs because despite the smallness of the cell, they are all separated one from another. And this separation is brought about by the surfaces and folds of the proteins, because they can make little pockets that are separate. That's one way. By complexes of the proteins fitting together, and by membrane-bounded organelles or non-membrane-bounded organelles. So these compartments, and this is an example of the different compartments you see in cells. Look at it in a different way. When we tend to look at this, we think it's very static and just describing the structure. I want you to look at it and remember these are the basis of thousands of different micro compartments which are necessary for this elaborate chemistry to take place. However, if it's all taking place independently of other chemical reactions, what do you have? Complete chaos. It cannot work. It has to be separate but linked together to function, to give you life. And that requires communication between the different compartments. And communication is the transfer of information. And that then leads to my next idea, that life is also, as well as being based on chemistry, it's based on information. And that's a very important point I'd like you to recognize. And it has various aspects. Life is a complex system. It's very intricately controlled. It behaves with a purpose to achieve certain things. And this is quite a complex concept. Now, life as a complex system was first suggested, you may be surprised, by a philosopher, Immanuel Kant, around 1800. And he argued, and we now would agree with this, that the operations of complex systems of life, of the systems of life, are based on these aspects and on the management of information. And I want to give you two examples. One, to illustrate this. One is the structure of DNA. We've already talked about that. That's why I put it up there. Now, the structure of DNA is iconic and beautiful. You know, we see it everywhere. But that isn't where the real beauty lies in the structure. The real beauty lies in the fact that it only makes sense when it's recognized that it's a digital information storage device. Back to what I described earlier. What you're looking at here is a digital information storage device, the one written in the linear script of 4 letters. So the linear polymer of DNA is ideal for storing information because all the encoding letter bases are turned inside the helix where they're protected. So it's very stable. But it is chemically inert, or more or less inert. So this information has to be turned into chemical action that can do work through the varied chemical activities of the encoded proteins. And that is a critical aspect of life which may not have been emphasized too much for you in your biology classes. It's a wonderful characteristic of life, the ability to encode information in inert linear structures and to carry out chemical and physical work in inactive 3-dimensional structures as well. The second example I want to talk about is this. This is a control, a regulation found in a man-made machine. It's easier to look at man-made machines to think about this than cells because you can see what they're actually doing. This is a governor developed by James Watt, who developed the steam engine, and it regulates a steam engine. And all of us can understand this. As the engine goes faster, those balls spin out. That lifts up a valve and shuts off steam to the engine. So it slows down, and then the balls come back in again, and that opens up the valve. Easy to understand. What it is an example of a negative feedback loop, which in biology is often applied to metabolic pathways. And here we have a simple one, the upper one. A is turned into B and turned into C. As C builds up, it inhibits A to B, and that leads to homeostasis. So that's a negative feedback loop, just like the governor. You can also have a positive feedback loop, which is the complete opposite. A goes to B to C, and C speeds up A to B, so this turns it into a switch. It's not homeostatic. You go from 0 to 1 with such a control. There's other control modules which were first discovered actually by 2 French geneticists Francois Jacob and Jacques Monod in the 1950s and 1960s, studying sugar metabolism in bacteria, is how they discovered that. But there's many such feedbacks and control modules in cells: homeostasis, generating homeostasis, switches, timers, toggles, oscillators, all translating chemistry into information modules. So that's the point I'm trying to make. The chemistry is turned into an information module to give, which when linked together can give elaborate routines of informational management. A metaphor for this is this. This is an electronic circuit. An electronic circuit, and it looks like a sort of a primitive radio here, in fact. And you see somebody's put the names of genes on there, actually genes all involved in cancer. They're connected to make a circuit. However, this is a bit misleading, just to make the point, because this is hardwired. It's connected by hard wires. Life is cleverer, because it connects the different components through solution chemistry, through chemistry, and you can rewire it. So as well as putting on top of it a different program, you can actually change the hardware as well. This is just to show the level of sophistication that living things can actually have. And that leads to a system that behaves as a whole with overall purpose, like reproducing a cell, which is what I do research in, making a butterfly, maintaining our cells. Now, for centuries, the generation of such purposeful behaviors was thought to require a designer, a creator. And it took evolution by natural selection to change this. And that's going to be my final idea. And when I've explained that, I'm going to put it all together, okay? So if you're getting a bit tired, take a big breath, deep breath. It's soon going to finish, and not much more to go. But this is my final idea. The most beautiful idea in biology is this. It's due mainly, not entirely, but mainly due to Charles Darwin, who was working in the 19th century. And it has two aspects. The first is that life evolves over time. And the second is that the major mechanism for that evolution is natural selection. So there's two aspects to it. The first of those ideas, that life evolves, was not Darwin's idea. It had been around for about a century before him. What Darwin did was put together much better evidence. I'm going to show you, on the right here, you see Charles Darwin, rather stern-looking 19th century gentleman. But I want to draw attention to his grandfather, who's the other person, Erasmus Darwin. Now, Erasmus Darwin, who lived 50, 60 years before Charles, was one of the scientist naturalists who talked about life evolving. There were others like Lamarck, for example, in France. Erasmus was very colourful. He lived in the Midlands, not far from Nuneaton, actually. I was talking to somebody who came from Nuneaton. And he was a colourful character. He was a scientist. He was a doctor. He was a poet. And he wrote up all his scientific studies in the form of poetry. I have a number of his books from the 18th century, and you read these things. I mean, it's really weird for us today. You read these poems describing his speculations and his experiments. He was also quite a radical politician. He was a Republican, so he wouldn't be the doctor for King George III, who was the king at the time. He was asked 2 or 3 times to do it, and he refused it, very unusual. He was a proponent of women's education. At the time, women did not go to any schools. He set up a women's school, and his 2 daughters acted as teachers there. And he was known to be a great conversationalist, despite having a terrible stammer. So he's a very interesting person. By the way, he also ate too much. And so he had a very large stomach, and he cut out an oval in his dining room table so he could sit comfortably next to his dinner. Anyway, that was just a bit of personal history about Erasmus Darwin. And by the way, he was one of the founders of the Lunar Society, which is one of the first scientific societies in the world that met once every month under the full moon. They were called the lunatics as well, by the way, to actually account for that. Anyway, now, they talked about the life evolving, and that therefore being that there is a tree of life. That is, that all living organisms are related. And if life only arose and survived once on the planet Earth, as seems likely, given the fact that the chemistry is very similar from a bacteria to us, then that basically means that all life on Earth is related to each other. So we are related to the humble bacterium or the humble yeast. It just puts us in perspective, but they are some of our relatives. I'll return to that in a moment. This connection was, this is the only illustration in Darwin's great book, The Origin of Species, published in 1859, where he drew this illustration of a tree of life. And you'll see there's a tree here, and that each of these are a different species, and some of them die out, like the dinosaurs, for example. And what you see at the top there is everything that has survived. Notice at the bottom, A to L, he wasn't sure whether life arose once or many times. We'd now probably say on at least the only life forms that have survived only arose actually once. Now, Charles Darwin, who drew this, gathered evidence in favor of this when he was on a trip around the world in the boat, the Beagle. He was 5 years on the boat. And he proposed natural selection, which is largely his idea. So that's his unique idea. And I'll just explain that very simply. It's a consequence of a population of species which exhibit variations. That is, they're not all exactly the same, the individuals within it. And that those variations are caused by inheritable genetic changes. And these can be caused by mistakes when DNA is copied or DNA is damaged, for example, and they influence the characteristics that make an organism more or less successful. And so over time, organisms which are better adapted to what they have to do reproduce more, and that variation spreads through the population. And that eventually leads to enough changes that individuals of the new population will no longer are able to breed with the original population. And so you have a new species, which accounts for the origin, the word, the origin of species, which is what he was trying to explain. He came to that conclusion partly by studies of finches' beaks on the Galapagos Islands. Now, if you look here on the left, you'll see different types of beaks in these finches. The Galapagos Islands are volcanic. They've only been around for a few millions of years, and there was probably a finch that got there. It's 1,000 miles from South America, that got there and diverged in this way, and you can see how adapted they are. The one at the top breaks nuts. The one at the bottom gets insects from the bark. See how beautifully adapted they are? On the right, you'll see what we have is different pliers. And these pliers are also adapted for different techniques. The one at the top is sort of like a wrench. The one at the bottom is much, much finer work. The difference is, of course, the one on the left occurred by evolution, by natural selection. The one on the right are intelligently designed. And this is the big divide of talking about intelligent design versus natural selection. Now, for natural selection to take place, you need three things. Reproduction, obviously. Without reproduction, it doesn't work. 2, a heredity system, because of what I've described to you. And 3, that heredity system needs to exhibit variability. If it was always constant, it wouldn't work. And that is exactly based on what I've already told you about cells and DNA, and I'm just going to describe it in this case. Imagine a single-celled organism. Imagine there's a gene in there which determines the color of the cell. So the cell could be yellow or it could be red. Imagine a mutation that turns a yellow cell into a red one. Also imagine that the red cell doesn't taste good to a predator. Then what will happen is the yellow cell population will turn into a red cell population. And that is an example of evolution by natural selection, a very simple one, but an example. This is a profoundly important and beautiful idea, so much so that the geneticist Hermann Muller in the 1960s used it to define life. And I'm going to go back to this in a moment. He said, living things have properties which allow them to undergo natural selection, therefore to evolve. Now, before I do that, I want to say goodbye to Charles Darwin. This is what he looked like as an old man with a big white beard. And this is the last sentence of The Origin of Species. And can I read to you, because it's one of the most beautiful sentences. Whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. I wish we wrote sentences like this today. I have to say, we don't. And you can still read this 150 years later, and it's inspiring. It's the last sentences of The Origin of Species. Notice also he referred to gravity. Now, the most famous scientist of the time was still Isaac Newton. He died 150 years before, because he produced laws of physics. And what Darwin was doing here is saying there are also laws of biology, because biology was very descriptive. You just describe all the variety, but there's actually principles, as I've been talking to you today, that underlie principles. And Darwin was saying there's a principle here that's very important, just like the principle of gravity that Isaac Newton was discussing. Now, I'm close to the end, and I'm going to give my summing up, but we have to talk about viruses, okay? Because are viruses alive or are they dead? And I'm going to give you an answer. Whether you'll find it satisfactory, I'm not so sure. But that is a critical question, because it's often what you're asked to discuss. Is a virus alive or is it dead? Now, what are viruses? Well, we all know about COVID, of course. Viruses have a genome, either DNA or RNA, because RNA can sometimes be heredity. They undergo evolution by natural selection, as we saw with the different COVID-19 variants. Thus, they pass the Muller's test that I have just explained. But they can only reproduce when they are inside the cells of other organisms. So does that mean they're alive, or does it mean they're not alive? That's the crucial question. And they operate in cells by hijacking the cell's machinery to copy itself, its virus, and to make more of its own virus. So this means a virus is completely dependent upon another living entity. So that's our problem. But the question is, are viruses so completely different from other living things? Because actually, all forms of life are often, to a greater or lesser extent, dependent on other living beings. I mean, the many parasites that live on or inside you, other animals or plants, would be examples of that. The extent of these dependencies are much less than it is for a virus, but you're still dependent. There are amino acids you cannot make, and you have to have bacteria in you making them. People sometimes forget that. So all living organisms are to some extent dependent upon other living organisms. So viruses they are an extreme case, but we have to realize there is a continuum, a graded spectrum of living organisms. So I offer this as as if you like something to think about when asked the question about a virus. It's at one end of a spectrum, which is extreme, but we are all dependent on other living organisms. And what this means is that life on Earth, at least, is fundamentally connected and being related as a result of evolution. So as well as being dependent on other living things, we're connected because we're related to other living organisms. Okay, now I'm just going to summarize where we've got to and then say a few words after that. What is life then? And I'm repeating some of the things I've said. It sums up. They are independent, bounded physical entities, and the basic unit of life on Earth, at least, is the cell. And that I explained before at my beginning. They're separated from the environment, they're in communication with it, and therefore they can generate order and organization, second law of thermodynamics, and multicellular organisms like you and me are aggregates of cells. 2, living things possess the ability to evolve by natural selection. That's Muller's point. And to evolve, as I've just explained, they have to be able to grow and reproduce, and they have to have a hereditary system which exhibits variability. 3, living things are chemical, physical, and informational machines, as I've explained. They construct their own metabolism. I didn't say that specifically, but that was clear. And that metabolism is used to maintain themselves, to grow, and to reproduce. Now, if you looked up a dictionary, this would be too long to see as a definition, which is one of the reasons why defining life has been so difficult. It is multifaceted. You have to consider many things. There's other aspects which are important for life on Earth, because what I just said could apply to life anywhere. Life on Earth is based on carbon polymer chemistry, but it doesn't have to be. You could imagine other systems. Difficult to imagine, but not impossible. They encode information in nucleic acid information storage devices, and they are protein, chemical, and physical machines. The informational management they have is on storage in a linear form encoded in the polymers, and all living things on Earth are related by common descent and are closely connected and dependent upon other living things. That takes us into the territory of ecology and the interactions of all living things. Now, should we ever encounter life elsewhere in the universe, and the universe is so vast that one suspects there will be, I think it will be built on similar principles, not necessarily the same chemistries, but similar principles. And the critical one, I think, is you need long-term information storage, and that, I think, requires chemistry based on polymers. And because we have the capability of storing information and also turning that information into chemical action. And you have to solve those two questions. Now, just to end, and to return back to our planet. Our planet's the only corner of the universe where we know for certain life exists. Okay, so we should hang on to that. It exists here. The life we are part of here on Earth is very surprising, but we are making sense of it. And the knowledge that we're getting shows us that living organisms we know of are related and closely interacting. And we are bound by this connectedness to all other life, to bacteria, fermenting yeast, which is what I work on, pea plants, the yellow butterfly I started this talk with. All these species are life's great survivors. They're the ones that still exist today as a consequence of evolution. But they are, and I said this already, the latest descendants of a single, immeasurably vast family lineage that stretches back through time, an unbroken chain of cell divisions that goes back to deep time. I want to emphasize that because there's a physical connection through reproduction and so on that just goes back forever. But as far as we know, we're the only life forms who, certainly on Earth, who can see this and reflect on what it means. And I argue this gives us human beings a really special responsibility for other life on this planet. You know, they're our relatives, is one way to think about it. They are made up of our relatives, some very close, like the apes, some ridiculously distant, but they are still our relatives. So we need to care about it, and we need to care for it. And to do that, we need to understand it, which is really why I've been talking to you. So thank you very much for listening to me. Thank you. Hello, who are you?
Hi, my name's Adwaita, and my question is that in the beginning you said that every cell comes from another cell, right?
Yes.
So how did the first cells appear? Like first cells
Well,
on Earth?
the question I'm being asked is, what is the origin of life?
Yeah.
And the short answer is, I don't know. Once you have a living cell, I can imagine everything else. How that cell came about is really quite a mystery. Came about, I can tell you a few facts. It came about on Earth about 3.7 billion years ago. We see the first fossils that we think are life. The Earth only cooled down about 4.2 billion years ago, 4.3 billion years ago. So within 500 million years of the Earth cooling down, we had cells that we now see as fossils. It's, of course, a long time, but rather short, in my view, for the most difficult part of evolution, because it went from, it took from 3.7 billion years ago to about 600 million years ago to go from a single cell to a multi-cell organism, which for me is trivial, a trivial transition compared with the first. It's a big mystery. Can't answer your question, sorry.
It's okay, thank you.
Hello, sir. My name's Adhit, and I have three questions, but
One
okay,
question,
one
please.
question.
Yep.
Okay, sure. There was a paper in the archive detecting sulfur dioxide on a planet, I believe, a few months ago. So, what do you think of that paper? Is it possible that other life forms reside on that planet? And also, if so, what kind of polymer chemistry base, as you put it, would be possible?
So I didn't catch all of that.
So he's asking about a paper which talked about the presence of sulfur dioxide. What did you say? Yeah. So he's asking about other forms of life and the chemicals which could actually result in life forms. Is
You
that your
mean, for
question? Yeah.
example, on the moons going around Saturn and Jupiter, were you thinking of? Yeah. So this is quite exciting because we used to think that the only other planet in our solar system that might have had life was Mars. No fossil evidence has emerged, and there's certainly nothing alive there now. There may have been billions of years ago, we don't know. But the discovery recently is that there's, just in case people don't know it, there are some moons going around Jupiter and Saturn, which were thought to be so far out they'd be too cold. But the geothermal energy within the moon has created enough heat for there to be liquid water. And liquid water is essential, obviously, for life. So this is quite exciting because it does provide an environment outside the Earth where one could imagine life evolving. So it's an interesting question because if we do discover that, will it be built in the same way as life on this planet? And that would really, if that was true, would suggest that life long time ago could be transferred across quite vast distances, at least within the solar system, and that maybe life on Earth was seeded by life from elsewhere. Now, that's within a solar system, which of course is hundreds of millions of miles, but that's a tiny distance to the next star. So it's difficult to imagine. You could imagine life coming on a comet or something, maybe within the solar system. But the nearest star is 4 light years away, I think. And I mean, that is such a vast distance, I can't imagine it. So it's very exciting. If we discover life in that situation, will it be the same as life that we have here? And if it's still alive, we can ask that question. If it's a fossil, that might be more difficult. Just to say, in case not everybody's familiar with it, there are gushes of water coming up out of that of these moons, and satellites have seen it, and we could design a satellite that could catch that water, and so then analyze it and see if there's life forms in it. So it is exciting. So good question.
I'm Vihan Swarankar, and my question was, how did the first multicellular organism come into being?
Sorry, I wasn't, again, I didn't, you don't mind repeating.
How did the first multicellular organism come into being?
Well, it's the same answer, I don't know. Sorry, it's exactly the same. I mean, that is the big mystery.
Hello, sir. I really liked this lecture, and I've been listening to the relationships between animals and the nature of symbiotic relationships as well. And you told that blue cells and red cells, that concept in which you said that
You're talking about algae, are you? The blue and red
no,
algae?
the predator prey. The predator prey concept you explained. So the blue cells change into red cells if the predator does not like the blue cells, the proteins in it. But does that mean that if we create a cycle between symbiotic relationships and predator prey relationship, that means the maximum symbiotic relationships that will survive over time in the tree would be located at the grassroots level or the first tropic level? Because as we see, a predator would not like prey, and that prey would be a predator for another prey, and like it would keep changing until a grassroots level, which does not be a predator, that are plants. So won't the symbiotic relationships be maximum there, and wouldn't we find the oldest symbiotic relationships at the grassroots?
Well, this is a long question. What you're describing is the complexity of relationships between living organisms. You had a particular emphasis on symbiosis, and symbiosis, I'm sure everybody probably knows here, is when a relationship between two organisms help each other. It's a sort of different to parasitism where one doesn't. Now, this, you see, we are to some extent all symbiotic in actual fact. It was a term that was used mainly for very closely connected, like you were mentioning, there are algae which have some fungi, for example, living in them. But in fact, you and a sheep are symbiotic, if you eat mutton, that is, okay? And there is a symbiotic relationship there as well. So when we are thinking about life, we have to think about this ecological complexity and the interdependence. What you're emphasizing is exactly the interdependence I was emphasizing, which in the end, taken to the extreme, ends up with the virus and provides you an answer to the virus. So ecology has a role to play in thinking about molecular explanations which we haven't fully realized before. Okay.
Thank you.
Yeah. Hi, sir. So you'd mentioned that life has basically evolved a sophisticated system over time. But currently, right now, how do we know that this is the pinnacle of sophistication when it comes to life? And if we don't know, what are like the speculations and theories for further evolution?
Well, again, it's an unanswerable question, knowing where life will evolve to. What we need to understand in thinking about those questions is that life evolves in changing circumstances in which it finds itself. And those changing circumstances can be interrelations with other life forms, which are very powerful, but also interactions with the environment, like the temperature or the level of humidity and other aspects of the environment. And these put very significant pressures on the selection of where organisms were evolved to. So we shouldn't forget that it wasn't so long ago that we had an ice age which covered much of the northern and southern hemispheres. And of course, all life had to adapt to that or simply not survive it. And so this is a situation that that life has experienced over millions of years. What is different today is the speed in which it is happening. And that is something that we need to take care of, because if it happens gradually, like an ice age, it changes so slowly that life forms have time to adapt and time to deal with it. If it occurs very rapidly, as it is now in consequence of human action, the one that we think of most is, of course, the increase in the temperature of the Earth because of burning of fossil fuels, life forms do not have the time to adapt, including ourselves, in actual fact. And so this is the reason why people like David Attenborough and so on, who TV naturalist, is always warning us about this. If we let the temperatures rise too quickly, we put tremendous pressure on other life forms. Now, we will survive, but we could be surviving very impoverished way with a lot of destruction. And what we need is political leadership who will actually deliver that, who look beyond 4 and 5 year election cycles and look and think about the future 50 and 100 years ago. So you see the nonsense with Trump at the moment, who simply denies this and is, I'm afraid, resulting in very backward pressure on the future of the Earth. And we have to really push back on that.
Thank you,
That
sir.
was a political message, by the way.
Thank you, sir.
So you kept saying that information is stored in a linear form in DNA. So what other form can it be stored in?
RNA, which is so similar to DNA, you'd say, is deoxyribonucleic acid is paired, so it's a helix, it's the ladder. RNA is not so strongly paired, so it's a single nucleic acid molecule. We do not store information in that way in a protein because it can't be turned into anything else. The thing about DNA is it can be turned into RNA and then turned into protein. When I say turned into, I mean the information can be turned into it. So that storage of information is physically encoded. But there's a more subtle answer to the question, which isn't quite the one that you asked, because it is that to understand living things, we have to really understand in a much fuller way how information is processed in living cells and organisms. And that requires a much more sophisticated thinking about how complex systems work. And information will be really important there, but it isn't quite the same thing as information storage. The short-term storage, which I can give you an example of, you can have a molecule which will be in a particular form, which will have a consequence, and it could be stored that way, but it's very limited information, not at all like the digital storage of nucleic acid. The reason I'm emphasizing that is that when people think in biology about information and the management of information, they tend just to think about DNA and that storage, whereas, as I think you might have been hinting, information is critical throughout the whole living system. It's just not the same sort of storage. Okay?
Thank you.
So, hi sir, I'm Vidya. So, I wanted to ask that you had mentioned something in the slide about viruses, and there's also I've noticed that in many places, like people are hesitating to they're hesitating to classify it in a specific kind of category. Like, for example, some people classify it as living. Some people classify it as non-living, but what I've observed is that viruses show more living characteristics than non-living. So could this affect like how we categorize it? Could we classify it as living?
Well, I tried to define, I think the question is built around the definition of life. Yeah. And it was central to what I was saying, but it isn't something you can say in a single sentence in a way. And so I ended up with this rather, 4 slides full of words. So, it might be best to consider this as like a description of the characteristics of life. For me, the most important principle there is the ability to evolve by natural selection. And that is where I would invest the most interest, okay? Then beyond that are peripheral things that are important for it. And if you say that, then a virus is alive, okay, without any doubt. If you focus a bit more on the chemistry and physics underlying life, then a virus would fall out of that. So because it relies on another organism. So to some extent, the answer of what is alive depends on how you think about it. And I have one preference, others would have another.
So I wanted to ask, it's that viruses are dead outside our body, but when they come into us and into the host, they come back to life.
Yeah.
But how is that possible? How, like, isn't that against nature itself for
Well-
the dead to come back to life?
Yes, in fact, in the book, I said some people say that a virus is dead outside, it's a chemical, and it's alive when it's inside the cell. So that I didn't mention that today. It's in my book, and that is another way of thinking about it. But it is a bit weird. I mean, to think of it. The reason people say that is because they are focusing a bit more on the chemistry, if you like. And a virus is like a chemical. When it's outside the body, you can crystallize it and so on. And so it's sort of, they're thinking of it as a chemical entity, which is not alive, whereas inside the cell, it has got purpose to replicate itself. You're looking as if you didn't like that answer. Did you?
No, I wanted to, like, I had an example. I recently learned about mimivirus, right?
Yeah.
And even when it's outside a host, if a smaller virus, like, takes it as its host, then the smaller virus is alive inside of it. So how can something be alive inside of something dead when that is also supposed to be dead, right? So is it possible that, I don't know, viruses are just hibernating when
Yeah.
they're outside?
Well, actually, it doesn't just apply, just to make it, to support you're basically emphasizing the complexity of this. There are other elements which have viral type properties. We think of viruses as an entity because they can survive outside a cell. But there are other DNA elements inside a cell, and this would be like Richard Dawkins' selfish DNA, which reproduce and undergo natural selection within a cell. So it's, if you like, an even more extreme case than the virus. So we have yet again, as comes up in life all the time, and it's the reason we have problems. We always want to, make taxonomic differences between these things. And you start with, a cell which is alive, you have a virus, and then you have DNA elements that can jump from one place in the genome or even between cells sometimes. So it all depends, like I just said, what emphasis you want to put on different parts. For me, the important principle, I'm going to repeat myself, is does it undergo evolution by natural selection? And that's what I'd go for.
Good morning, sir. I'm Athar. I want to ask you, how can someone like us work on, start researching and to have some impact on science society, to work on something which is very noble?
I'm often asked, and people do ask, where is research going? And I always answer, I do not know. Okay, it's the same answer. By the way, you've got to be honest. When you don't know, you should say you don't know. Otherwise you get into bullshit. I mean, it's just not. It's just better to be honest about it. Now, if I could predict where it was going, it's probably a boring prediction. And so I get irritated with books that are always looking over the horizon for the next great discovery, because usually they don't happen. Actually, my book is boring because you'll still be able to read it in 30 years and it will be correct, okay? Because there's nothing in it which is, very little that is likely to be wrong. When people ask that question, though, they are often thinking of applications of science rather than science itself. And the reason I mentioned it in this way is to make that clarity of a distinction. Because let's take something like gene editing, where you can change the structure of genes. That wasn't invented recently, actually. It was invented 30, 40 years ago. I did it for yeast, for example. But it's only been done efficiently for example, human beings or animals or plants in the last 10 or so years. So now you can think, well, now we can do that. What can we do with it? So we could make plants that might make, bigger grain. We can produce different ways of treating diseases and so on. And those applications you can predict, and that's what happens in the commercial sphere, where discoveries are turned into application. But discoveries are discoveries. What it means is it's not actually known at this point. And so by almost its definition, you don't know what it is. I started working, as you heard, on the cell cycle when nobody was in the least bit interested in it. And I only fell into it by accident, really. And that is really often how discoveries start. Something unknown emerges. And so predicting discoveries is really quite difficult. You have, well, a whole different talk could be on how you looking into the future is very difficult, and you have to develop your imagination, develop your curiosity, and follow things in different ways from what people have done in the past. And this is why I emphasize it, because we tend, when we ask that question, to look at the past and think how that is going to go to the future, whereas actually real discoveries are usually a break in the past. Yeah?
I was just wondering, where did viruses come from? Like, did they evolve from the other living stuff? Because wouldn't that make them alive? And if they didn't, couldn't they, like, later on evolve to become alive? Definitely.
Viruses have been around a very long time. And when you ask how they came into being, we can only sort of speculate for the origins. But essentially, a virus has got a piece of DNA or RNA in it that makes something, usually a coat for itself. And almost certainly its origins were in the genome of a plant, animal, or a fungus that turned itself into a virus. So I think that's probably the origins, but they've been around for so long that it's a bit difficult to answer the question.
Sir, I wanted to ask, how do we know that extraterrestrial life will be at all similar to us? Like, how do we know that they can't bear extreme temperatures or that they would need variation?
Well, it's an impossible question because we don't even know what extraterrestrial life is out there yet and what form it takes. So you could imagine, it's best to read science fiction novels if you want an answer to that question in a way, because we don't know what life forms will be out there. So we can't really describe it. If you followed the principles I was talking about, then if they are correct, then we could imagine some form that they might take. But until we find it, we can't even think about the question. Yeah?
Hari Om sir, my name is Yashvardhan. So your session was really good. I got some great insights. My question was, sir, as you give the example for the knuckles, like how it was so similar to a bird's beak, isn't the way that DNA is coded right now, you said like it acts as a building blocks of information, and it's a great way of storing information, and it's the most efficient way. Then why aren't we using these in our computing systems? Why isn't there any way of us replicating or making a code which uses the same structure as artificial DNA? Because that will be the most efficient way. And especially with the growing advancements in artificial intelligence, we should replicate it that way so we can create something similar to us, right?
I think you're talking about the ability to artificially make DNA, and does that lead to differences from... So this is an interesting question. We understand how DNA works. We understand how it's transcribed and translated into proteins. Up until now, we've mainly thought about variations on pre-existing proteins, trying to modify them. But actually, proteins as we know them now occupy what is called a limited chemical space. And we can imagine a much bigger chemical space if we designed it ourselves. We simply don't know how to design it. Now, using some methods of artificial intelligence, you can imagine other aspects of chemical space which you could perhaps occupy, that you could move to. Now, if that works, and I would say at the moment it is on the edge, but it's not worked well. When people do things like this, they always say it's solved and so on, but it's not. But if that does work, then the question you're asking does have real potential. The reason I am wary of it is there are reasons why chemical space is restricted, which is that it works, and that it is possible that when you go into other chemical spaces, although you can imagine it, it won't actually be functional. So it all depends on whether life is evolved into a state where it doesn't allow certain sorts of variants, which limits the chemical space, or whether we can actually design cells or chemical ways of making things that can occupy different chemical space. So it's a very important question which we're talking about, but really just scratching the surface. I don't know if I've answered your question. Thank you.
Sir, I was reading about this theory that the first cell that was formed on Earth was a bacteria cell. So the DNA like that was present in it, didn't had a nucleus and all. So the DNA that was there is configured and is fitting the bacteria cell. Then there was like, for evolution to happen and variation to happen, isn't it like necessary for something to be present in that thing? Like for finches, they all had beaks. Variation occurred when there were like small differences that led to big change in the long run. But if you never had a nucleus, how did the present beings evolve?
Okay, the question is a bit complicated. So her question is, how is something evolving if it doesn't have a nucleus? For example, you're talking about finches and then it has beaks, other features.
Yeah.
But how does it happen when you are so, there's no nucleus inside or nothing which dictates evolution?
Well, it is a complicated question, but we have to remember that the nucleus is just a way of packaging the DNA in certain structures. And most life that we see, all of it that we look at, does have a nucleus. Bacteria don't have a typical nucleus like that. So that's already a different sort of living organism that doesn't have, they call it a nucleoid, because it has some nuclear features, but it doesn't, it's not exactly the same. We could easily imagine life with no nucleus, just having DNA. That I don't think would be a problem. It may not be efficient or effective, but we could easily imagine it. And of course, as you were probably thinking, a virus, for example, has no nucleus. It just has a coat of protein. So for me, it's not such a big problem. You could imagine it, but it's probably not efficient or effective, and so it's been evolved away. So if we were to transfer ourselves back to 3 billion years ago, indeed, there may have been organisms that have no nucleus at all, but it is turned out to be more efficient, effective, and evolved to have a nucleus to do those things. I'm only making it up because we don't know what happened 3 billion years ago. But I do not think it is such a big barrier not to have a nucleus. Yes, so you're saying, so did the cell membrane have to come from somewhere? So it's an even bigger problem. As I said over there, I do not know how first life forms form, because you have to have a membrane round a cell. I think once you've got a membrane around the cell, I could easily imagine a nucleus, because you could then pinch the membrane off. So I would re-couch your question to thinking, how did a cell surrounded by a membrane come about? And my answer was, I don't know.